Now we're moving into Chapter 6, Part 4. We're going to wrap up some cytoskeletal elements, talk a little bit about cell walls, and complete this chapter. Big chapter, I know. I'm going to start off with microfilaments, which are the smallest cytoskeletal elements made up of actin monomers. We call those G-actin. And when they assemble into this sort of torsional fiber, if you will, they call that F-actin. Very, very good at holding tension, not very good at compression. So if you tried to squeeze it together like an accordion, it wouldn't hold up. But if you pull it tight, like a tight wire, it works quite well. It's designed to bear tension rather than compression. And you can see actin filaments in a mitosing cell during cytokinesis. You can see actin fibers all and around the what they call the cortical area of a cell. Those are stress fibers. And then we see actin filaments, microfilaments in skeletal and contractile muscle. So they can form 3D networks, or they can be very, very regimented, like in muscle. And they also are involved in things like microvilli and absorption structures in intestines. And so here are some of those microvilli, and the microfilaments are in there to provide some additional rigidity to those villi. Down here, they actually have different filaments. These are intermediate filaments, primarily keratin, whereas microfilaments are actin. Here's some of that cortical actin I was talking about. What we see here in green are microtubules, and what we're seeing in red are actin filaments, and you can see some of those stress fibers right there. They can help with shape, but they can also help with motility. They interact with motor proteins, particularly myosin, the motor protein myosin. And they don't have to be regimented. They can actually be in muscle fibers they are. But in smooth muscle and other tissues, they aren't arranged in that parallel shape. But myosin is thicker. It's got catalytic heads. Actin is really just a filament. Here are those regimented fibers I was talking about, and you see these real dark areas. That's where actin and myosin are overlapping. The actin filaments by themselves are found in this area. So these Z discs are where actin is anchored, and they extend out. And what happens is myosin will pull these Z discs toward each other. So they're pulling the actin into the myosin. And here's that arrangement. And here's one of those Z discs I was talking about. And here's another one. It's pulling those Z discs together. But actin doesn't have to be this regimented. It can be like in smooth muscle, you don't see it. That's why it's not striated. But you can also see it in monocellular organisms like this amoeba. When they stick these pseudopodia out, the actin actually dissolves and allows for the pseudopodia to stream as it does. And then the cell will stream into that pseudopodium. We can also see movement that we call cytoplasmic streaming. Actin is involved in the circulation of chloroplasts in the cytoplasmic streaming that we see inside certain types of plant cells. Here's a nice example of that cytoplasmic pseudopodia motion seen in an amoeba. So what you're doing is you're actually breaking down filamentous actin into globular actin, G-actin, and then that reduces the rigidity of the cell, and the cytoplasm can stream into that area. And here's a nice view of cytoplasmic streaming in what's called an Elodea. That's a type of plant we often use to observe plant cell function. So you can see that movement there. That's caused by actin and myosin interactions that then cause the cytoplasm to stream. And we have intermediate filaments, which are in between microfilaments and microtubules. and are the least understood and from what we understand, the least dynamic. They change less than the other two types of cytoskeletal elements. So right now, yeah, they support cell shape. They can help hold organelles into place, and you can actually see some tiny organelles in this beautiful image of intermediate filaments. This one over here is pretty cool too. What they've done is they've stained for the protein keratin. And keratin is one of the most common proteins found in intermediate filaments. Like I said, they're more permanent, less dynamic, more about shape, more about keeping things in an appropriate three-dimensional position rather than moving them around or something. Now we're going to transition into what we call extracellular components. What we're talking about are cell walls, extracellular matrix, and intracellular junctions. So cell walls we often know about in plants, but we also find them in fungi and in bacteria. The extracellular matrix we've talked about a little bit before. Lots of carbohydrates, proteins that are conjugated to them, like for instance proteoglycans are a combination of protein and carbohydrate, and other proteins in there. And it depends on the tissue, depends on the function of the tissue that drives what kind of material we have in the extracellular matrix. And then we have intracellular junctions, and we have three major ones: tight junctions, desmosomes, and what are called adherens junctions or communicating junctions. I'm going to start with cell walls, and we'll focus on plant cell walls. Main thing is that it's an extracellular structure. It's outside the plasma membrane, and it's relatively rigid, especially when compared to the plasma membrane. Now, cell walls can vary. You have non-woody plants and you have woody plants. It's just a matter of how much certain protein concentrations, namely a molecule called pectin. But in any case, it's an extracellular structure. Animal cells, by and large, do not have cell walls. But we do see them in bacteria, fungi, and in those sort of animal plant-like in-between things we call protists. They protect the cell, and most plant cells cannot survive, or at least can't thrive very well without a cell wall. You can strip a cell wall off of a plant in the lab, and it can sort of go, but it's really not. The plant cells don't do well without them. It also is a protective barrier to prevent plant cells from swelling when they are in hypotonic situations or hypoosmotic situations where there's a lot of water around. The cells can fill up in the constraints of the cell wall without bursting. If you were to do that with a human cell, they wouldn't last long in relatively dilute water. Made up of cellulose, but also other proteins and other polysaccharides. So cellulose is one of the main components, but things like pectins and other types of glycoproteins and so forth can contribute and make different cell walls in different plant tissues and in different plants. This is the generic structure, if you will. You've got a sort of primary cell wall that's actually in the middle. It's a little bit confusing because they have this middle lamella, which is on one side, and that's because this would be in between two cell walls. So they were looking at it in the context of multiple cells rather than just one. So there's this primary cell wall, there's the middle lamella that separates it from another cell wall, and then you have the plasma membrane here. And that's the structure that you see here, just in a slightly different image. Again, those big, the pectins kind of hold cellulose together. Cellulose are these big, large fibers, which are sort of the bulwark of that primary cell wall. Middle lamella will also contain different types of proteins, including pectin and others. And then you have your plasma membrane. You also have what are called primary cell walls, okay? And then you have, that's what we've already seen here. Then there's this secondary cell wall, which is hard to see in a picture. I may have another one here in a second. These cell walls do have holes in them, and we call those holes Plasmodesmata. It allows for osmotic sharing and cellular communication across these cell walls. Here's a figure from your book showing you two cell walls next to each other. So there are this darker layer, that's the middle lamella. Then you have the primary cell wall, that's going to be that cellulose embedded material. Then there's this secondary cell wall, which we're not going to talk about all that much. There's more to this cell wall, but again, the main thing is that its function. We're not going to dissect the cell wall all too much. Mostly what we're interested in is function. And speaking of function, here are some functional structures, the Plasmodesmata that I mentioned in the previous slide. You might want to take a look at the experiment that they talk about, of which this is the result in Figure 629. You might want to just take a quick peek at that in your book. It's showing that you have cellulose synthase enzyme and you have microtubules, and that the cellulose synthase seems to run identical. Like, for instance, let's look at this arrow here and this arrow here. That's a pretty obvious band, and then here and here. So what it looks like is that the cellulose synthase is being transported or otherwise is associated with microtubules when it's laying down cellulose. And while animal cells don't have a formal cell wall, nevertheless, there are structures in human tissue that have a rigidity and at least a sort of protein structure that might, give rise to someone saying, "Hey, that looks a little bit like a cell wall" from time to time. But generally, no, you don't have that. You don't have that formal cell wall. But we do have this extracellular matrix, and the components of extracellular matrix, proteins like collagen are very, very common. If cellulose is a common rigid fiber or semi-rigid fiber in plants, collagen is certainly a bulwark fiber in animal cells, and lots of proteoglycans, that is to say protein, carbohydrate, molecules that have proteins that have been post-translationally modified with carbohydrates, and there are a ton of carbohydrates. Carbohydrates are predominant out in the extracellular area. Even molecules that have been modified with carbohydrates are targeted often to the extracellular surface or for secretion. Fibronectin is another common protein that we find in the extracellular matrix. And again, it depends on the function of the tissue. Some extracellular matrix is meant for tension, like for instance, tendons and ligaments are very, very good for tension, but cartilage and other types of cushioning tissues have a different type of extracellular matrix, again, designed to absorb pressure rather than absorb tension. ECM proteins are also very, very important for letting cells know that they are in the right place. A normal cell, a normal animal cell, it has a default pathway of dying unless molecules like integrins, which are proteins embedded in the plasma membrane and are receptors, if those integrins are not stimulated, the cell will actually undergo cell death, apoptosis. But if those receptors are stimulated, they will send signals to the cell, to effectors in the cell, to say, hey, don't undergo apoptosis, survive. And so integrins are important protein molecule receptors that help the cell identify that it's in its right place and it's adjacent to extracellular matrix. Here's just another picture of that. Here are these integrin proteins that are a little more accurate from a drawing standpoint. They serve signaling and attachment function, these integrin proteins. So here's the extracellular fluid. Again, all these little carbohydrates, proteoglycans. We've got collagen in here. There's a fibronectin. There's one right here. Fibronectin hooking up to one of these integrin proteins. So these proteins serve linking and structural and signaling purposes. This is a fabulous slide. It shows you the basic functions of the extracellular matrix and examples of each of those functions. This is a wonderful slide. I would flag this for understanding and questions related to assignments or exams. If you have single cell organisms that work together, or single cell organisms that depend on knowing how many of other cells there are around them, or especially multicellular organisms, communication becomes critical to life. Let me just give you an example before I delve into this slide. The bacteria in your mouth can sense when there are a lot of them around or when there aren't a lot of them around. So when there aren't a lot of them around, they will undergo a certain level of metabolic activity. But when there are a lot of bacteria around, they will sense it. They call it quorum sensing, and they will shift their metabolism to a much more acidic byproduct and contribute to tooth decay. It's because you have lots and lots of cells. They can sense the quorum and they'll shift their metabolism because of it. And that's done in part because of cell junctions. So they allow for neighboring cells. They can communicate, they can collaborate, and they can sense when the presence or absence of neighboring cells. And so we have these junctions, and we're going to break some of these things out. We have three major ones: tight junctions, desmosomes, and what we call communicating junctions or gap junctions. To be sure, there are other ones. We have adherens junctions and the like, but these are the three that we're going to cover in this class. And here's a picture of each, and they can often be present on the same patch of cell membrane. Tight junctions are part and parcel of epithelial tissue. Very, very important semi-regulatory junctions. And what do I mean by that? Is they can be very, very tight and prevent anything from moving between the cells. We call that paracellular transport, when material can move from down here, say an intracellular area to an apical surface like a lumen of an organ. When material slides between the cells but not actually entering the cell, we call that paracellular, and tight junctions regulate that. Depending on what proteins are in that tight junction, maybe water gets through, but other things don't. The inside of your mouth and the inside of your airways allows fluid in. That's why the inside of your mouth is typically moist, is because the tight junctions can allow a little bit of water and some solute to creep through. But there are watertight junctions in other places like your large intestine and so forth. So tight junctions is really, really important that way. They're semi-regulatory. Desmosomes and then gap junctions. And then in plants we have what are called Plasmodesmata, which are basically like gap junctions, but they cross through cell walls. And so here's a picture of a Plasmodesmata right here. Okay, so let's break some of these out a little bit. Plasmodesmata, like I said, kind of function like gap junctions, only they're penetrating cell walls and not necessarily plasma membranes. And here's a lovely electron microscope image of some Plasmodesmata that are in this particular cell wall. So we're just going to show you some animations of each of these things. Like I said, tight junctions, they are often found in epithelial cells. They're usually kind of up high, kind of near the luminal surface of an epithelium. And they can be, like I said, semi-regulatory, or maybe I should just say it, regulatory. Like you can see these blue and green molecules in the squares, they're not getting through. Blue, I presume, is water, but some tight junctions do allow water, particularly in your airways and in your digestive tract, depending on what proteins are present in that tight junction. But if you've heard of the disease cholera, right? Cholera? Well, cholera attacks large intestinal cells with a cholera toxin, and that cholera toxin basically renders the tight junctions between the colonic cells, the colonic epithelial cells. It doesn't break them down, but it negates the barrier function, and so water can fly out of the intracellular spaces back into the intestinal tract, and people die of dehydration. They die of dehydration because the colon is fundamentally, the large intestine is fundamentally a desiccating organ. It's meant to pull water out of the remaining food material because of the fact that we live on land and water is sometimes relatively scarce. Gap junctions, we call those communicating junctions. They are pores in plasma membranes. They're special protein pores that create a space where ions, particularly calcium. Calcium's the really important one. Maybe water can get through there as well. Doesn't surprise me, but calcium's a big one, and it allows for cytoplasmic signaling from cell to cell. If this cell on the left releases some calcium in response to some sort of a stimulus, that calcium can not only cause effector proteins to turn on and off over here, that calcium will move through and affect effector proteins in another cell. Smooth muscle does this, and cardiac muscle does this. Because the last thing you want in cardiac muscle is a bunch of muscle fibers acting independently. You want them to contract as a unit. And so that's why they call this a communicating junction. And then lastly, desmosomes. These ones are really just anchoring junctions. There are also adherens junctions that are kind of in the desmosome category. They have these protein anchors, and they really are designed to facilitate strength of the sheet type epithelial tissue or connective tissue. Your pericardium, the outer layer of your pericardium, or the dura mater in your central nervous system around your brain, have lots and lots of desmosomes, and those sheets of epithelia and connective tissue are very, very strong, very, very durable. And just another picture here from showing the caricature, but also showing these things in electron microscope images. Wrapping up chapter 6, they want to talk a little bit about the idea of emergent properties in the cell. A cell is more than the sum of its parts. It has dynamic responsiveness. Even cells that are dead, which is really, really interesting, cells that are considered dead still have some responsiveness in some ways that is more than just the sum of its parts. And so the book talks about that. Just give it a quick read over. Not the most important part of the chapter, but nevertheless, it's good to reflect. It's good to reflect because most of you are aspiring to move on in the biological sciences or healthcare fields. It's good to understand that sometimes there are things we can't predict, and getting used to that fact, getting used to not knowing the full story is part of professional competency, and being comfortable with that and saying, hey, I might not know everything, but I can certainly manage and learn new things and manage what I don't know. And that concludes chapter 6. Thanks for hanging with me. Good luck with everything, and as always, please ask questions. I'm always delighted to hear your questions, help you understand this material better. Thanks again, and I will talk to you soon.